has to be validated by means of performing that binding or like we say agonist or antagonist of some particular receptor or some inhibitor of some enzyme or activator of some enzyme. So that target has to be validated by producing that action of that drug by different techniques are there. So for each and every step there is a detailed studies and require manpower. But I'll be going into the broad overview like how the drug development process takes place because every field requires a specialized expertise and specialized techniques and there's a lot of detail to be discussed under each area. But anyhow, studying if we look at a broadly the stages that include first identifying the research target, then validating the target, then after validating the target you have to design design the molecules by use of or help of computer aided drug designing, then synthesizing and carrying out HTS, that is high throughput screening, and identifying the lead identity molecules, and those lead molecules are then optimized with respect to use as a drug, and then preclinical toxicity studies are carried out for that molecule, and afterwards the important stage starts with this giving the dose or a drug first time in humans, a new drug which has never been given to humans, then that has to be given. But for that, you need to file an application called as IND. That is, filing an IND is investigational new drug application, which has to be filed with the regulatory authorities, especially like in case of India, it is CDSCO, Central Drug Standards Control Organization, and in case of US, it is US FDA, and similarly in specific the respective countries they have respective regulatory authorities. Then once you file an IND, you get a full from
Excuse
the
me,
regulatory
sir.
authority. Pardon? I'm audible?
Excuse me, sir. Hello.
Yes, yes, yes.
Sir, actually, are you sharing any screen right now?
Yeah.
Yeah, you are audible, sir. But not your screen, sir. We can't see your slides.
Okay. I asked you earlier that you can. Can you see it now?
Yes, sir.
Yes,
Yes, sir.
sir. It is visible now.
It is, it is there.
Okay, okay. Is it visual now?
Yes, sir. Actually, sir, you have to do it to full screen. Make it
Yeah,
to full screen.
it is a full screen now. Is it visible? Is it visible or not?
Yes, sir. It is visible.
Yes, sir.
Pardon now?
Yes, sir. It is visible. Sir, it is quite visible.
Is it visible?
Yes, sir. Yes, sir. Sir, it is visible.
Okay, okay. So I was talking about the stages of drug development. After filing the IND, once you get an approval from the regulatory authorities, regarding the permission for carrying out the clinical trials, then you have to carry out a clinical trial, which are divided to various phases as clinical phase 1, phase 2, phase 3. Then afterwards you file an application. Once there is successful conduct of phase 3, you file an application called as NDA. Earlier it was IND. Now it is called as the NDA, that is new drug application. Earlier it is investigational new drug application. Now it is new drug application because investigations are being carried out. And once this application is filed, the regulatory authorities they examine the application and decide accordingly. They either grant the approval or disapprove the drug. So if it is successful, they will grant the approval and the drug can be marketed. And after getting the approval, the drug has to be monitored for 2 to 5 years in the market, called as this post-marketing surveillance stage or post-marketing post-sales marketing phase by different names. Has the slide changed? Is the slide change visible?
Sir, the slide haven't changed.
It has changed?
No, sir. No.
Hasn't changed?
Yes, sir. Now it is there.
I,
Now it is there.
I don't know. There's maybe some issue of this. Anyhow, please let me know if there is no change in the slide at any moment.
Sure, sir.
Okay. So when we talk of these stages of drug development, it includes NCE, that is new chemical entity. A new chemical entity will be discovered and developed in the world which has not existed till now. Then afterwards includes preclinical studies, IND, ND, and all this told earlier. So when we talk of discovering a new chemical entity, how do we discover a first new entity? So first you identify a target. As I told earlier, you have to identify a target by using various techniques like bioinformatics. Then you have to validate the target by using techniques like genomics and development of that assay. For that validation of a target, you have to develop the assay for some particular receptor, enzyme, or ions, and uh, that will lead to target identification, validation, biological attributes, genetic information, and protein characters. So, like how the targets are identified, is the slide changing? Pardon, is the slide changing?
No, sir.
No.
No, sir. Yes, sir. Now it is there.
Shall I
Now,
move? Is it moving?
yes, sir. Yes, sir. Now it is there.
When I go into the slideshow mode, I think there is some issue.
Okay, okay.
So when I'm printing the slide, it's not moving. I don't know what's the issue. So like, how do you do it? By designing a target, designing a molecule by superimposing on it target structure. That is, the drugs usually act on cellular or genetic chemicals in the body known as targets. So they can be a cellular structure, genetic body chemicals. So identification and isolation of targets to learn more about their functions by the techniques called as bioinformatics, genomics, and proteomics. They are all involved, and then the target is identified and validated, and the compounds are identified or designed to interact on these targets. Some other statistics. Is the slide changing? Change? No?
No, sir. No.
So I don't know what the issue is there when
So now it is there. Now it is there.
pardon?
So it has changed.
So I don't understand. Like when I'm going to slideshow mode, it's moving on my side, but it's not displayed. I don't know where's the technical issue. Can I present it in this mode? Is it acceptable?
Yes, sir. Yes, sir. It is fine. Sir, you can go to the full screen. You can go to the F5 and Make it to full screen.
Full screen means that is slideshow, no?
Yes, sir. You have to go to the F5. F5, and
Yeah,
that is the
I'm doing
slide.
that. I'm doing F5, but when I'm moving the slides, then the next slide is not moving in that mode.
Okay.
That's the issue. I think I will try to manage in this way
Okay, fine, sir.
because if I go in the F5 and when I'm moving, it's not moving at your end. Well, in between there's some issue I feel.
Sir, this is fine.
If you want, I can move like that. Is it moving?
No, sir. Now it
Not
is not
moving?
moving.
Yeah, that's the issue I think.
Now it is moving, sir.
So then let me present in this mode itself. Is it manageable like this?
Yes, sir. It is visible.
So somehow we have to accommodate because if I am going in the slideshow mode, when I move the slides, there is some issue that it does not move at your end. Means in between there is some network or whatever issue may be there. So I think if it's acceptable, then I can manage like that. Current in this mode. So when we talk of this thing, molecules, once the target is identified and the molecules are synthesized, okay. So once the structure of a new compound is designed and synthesized, it is not one or two compounds, it is thousands of compounds which are designed and synthesized. So for the participants, it's a very important information when we talk why it takes so much time to discover a new drug or develop a new drug. Is basically it is an enormous work involving lot many number of permutation and combination of designing and synthesizing various molecules, thousands of molecules, and out of those thousands of molecules, they are then tried for their activity. And once they are tried for their activity, earlier it was done manually one by one molecule, but now there are advanced techniques available to analyze them, or we can call it as to screen them for identifying them as hits and leads. So when we have thousands of molecules, we have to save time and have increased output to filter out or screen out which are the effective compounds. Out of those thousands of molecules, a very few molecules will be having an possibility of activity. So those molecules which are showing some activity or which are able to have potential candidates are called as hits. There may be few in number, maybe 10, 20, or 30, something like that, out of thousands. Then out of those, again they are refined and examined or analyzed in detail to focus on some compounds like leads. These are called as leads. That is, those compounds out of the hits which are further selected for testing or development, and they are very few number, maybe two or three molecules, a very few single digit number in molecules. So they are then developed in depth, right? So this is a diagram or this is a figure which shows like there will be some hits will be identified. Out of those hits, a very few molecules will be identified as lead molecules, and these lead molecules will be optimized as as clinical candidates for development of a new drug. So once these lead molecules are identified they need to be optimized. That means optimized means they have to be carried out various types of steps are taken on that or optimized to develop or refine its properties, especially the physical and chemical properties, wherever the refinement can be done. So for optimizing that, the compounds are the properties are optimized, modified so that they have appropriate activity. So the selected compounds with greatest potential are to be developed into safe and effective medicine. So assessing the various functional groups by using analytical techniques which are responsible for efficacy, binding, or which are responsible for toxicity. So those all molecular functional groups are refined and optimized. So once this new chemical entity is formed, incised, and optimized, then you have to examine its efficacy and safety. So once the molecule, we have few molecules, maybe two or three molecules, or few in number, then they have to be evaluated for their safety, efficacy, and safety. So for carrying out those safety and efficacy, you have to perform various studies, which includes preclinical and clinical studies, broadly speaking. But primarily speaking, we have to focus, or first we have to test it on preclinical studies, or that is on experimental animals. So it becomes necessary to assess all these parameters before conducting any human clinical trial, assessing the safety, toxicity, kinetics, and metabolism. So the preclinical toxicities studies include
Sir, you cannot be heard.
Yes, sir. I think there's an audio problem.
Sort of, can you hear, sir? Sort of, can you hear, sir?
No, sir. It's not sir audible.
Okay, he actually left out for the meeting, so we have to wait for a few time. So he will be joining again. Maybe some technical glitches.
Yes, sir. Yes, sir.
So I should request all the participants to hold your patience and to be on the line. Sir is trying to come in. there are some technical problems, so it should be soon resolved, and sir will join the line. Please be online.
Yes, pardon, is it my screen visible? Is my screen and audio, am I audible and visible?
Yes, sir, audible, sir.
Okay, thank you, dear.
Audible, sir.
Okay, thank you. Sorry for the interruption due to network issue.
Okay, sir.
So I'll just go back till here. Was it okay? After carrying out the clinical studies, drugs that pass the initial screening and profiling are carefully evaluated for potential risk before and during clinical trials. Preclinical studies include safety and toxicity testing, generally conducted on animals, microbes, and tissue cultures. So, as I'm telling you that the regulatory authorities, they are more concerned about the safety issues, rather than the efficacy. No doubt, efficacy is required, as I told, but the focus or the more stress is upon the toxicity studies. So, toxicity studies, various types of toxicity studies are carried out, which include acute toxicity studies, subacute and chronic toxicity studies. What does it include is, in the acute toxicity studies, the drug is given in a single dose. One single dose of drug is given as per the OECD guidelines. There is a guidelines called as OECD guidelines, guideline number 421. Okay, so in that, a step-by-step increasing dose is used. We start with a 2000 mg dose, whether it is fatal or not. Depending on that, the drug is increased or decreased. If the 2000 mg dose is fatal, the drug is decreased down, or the dose is decreased down and examined. So likewise, the acute toxicity studies are carried out. In this, single dose is given, but however, the drug is monitored, or the effect on the animals is monitored for 14 days, and that is single dose drug is given to two species of animals, and those two species include basically is one rodent species and one mammalian species, and you have to use two routes of administration for every species and three dose levels for every different route and different species. So you can imagine the number of animals or amount of work increases. Two species of animals, in each species you have to use two routes. That is, two routes means one is the intended route. We call it as intended route of administration. That is, which has to be given in the humans, maybe oral, parenteral, or whatever it is, and any other one more route. And you have to use or study three dose levels as low, intermediate, and high dose levels for every route and species. And you have to monitor it for 14 days for various parameters, which include behavioral parameters, physiological parameters, biochemical parameters, hematological parameters. And after that, 14 days evaluating all the parameters, you have to sacrifice the animals, all animals, even the rodents, mammals, mammalian species includes dogs and monkeys. You have to sacrifice all the animals and take out various organs. There is a list of organs to be taken out, which include more than 30 organs. 30 or 32 organs are there, which has to be taken out for all animals. Their macroscopy examination, microscopic examination, histopathological studies, all those has to be carried out. And what are changes in the toxicity in the organs. Similarly, for subacute and chronic, the studies are carried out the same way for biochemical, physiological, hematological, and histopathological changes, but the duration is different. That it varies from 1 to 12 months. Why I have mentioned it 1 to 12 months is it varies from country to country. In some countries, the subacute ranges from 1 to the subacute ranges from 1 to 3 months, and in some countries subacute ranges from 1 to 6 months. Variable time period for different country to country, and chronic toxicity also varies from country to country. Like in some countries it is having norms of 1 to 9 months, in some countries it is 1 to 12 months, and in some countries it is even more also. So the purpose of chronic toxicity is to see if a drug is given for a longer time, like the drugs like anti-cancer drugs or anti-diabetic drugs, anti-hypertensive drugs, which are to be taken for long duration or lifelong. Then they are to be examined for their toxicity. That's why we use the term as acute, subacute, and chronic toxicity. So I hope this will be clear. Then there are studies for toxicity carried out are reproductive toxicity studies, carcinogenicity studies, mutagenicity studies, dermatological toxicity, and
Okay, sir, the audible.
for the reproductive toxicity studies means in this the in the reproductive toxicity studies the effect on reproductive function is studied, including teratogenicity and postnatal developments. Now see these are just two lines. We can if we just see these appears to be only two lines: effect on reproductive function and effect on teratogenic potential and postnatal development. If you see, it includes three components. That is, one is effect on the reproductive system of animals. Then, what is the effect of reproductive toxicity on the teratogenic potential? That is the embryo and fetus that are delivered. What is the effect on them? And next is postnatal development, means when they get mature, the embryo, fetus, or the that delivered the next generation animals, when they become mature, are they able to reproduce or not? You have to wait till their maturity and becoming reproductively mature and delivering one more generation. So you can think of it, you have to wait for two generations to be delivered. So that's a very important point. That's why it's a time-taking process. The drug development process, why it takes so much time, is you have to wait for two generations, and the animal requirement is same as earlier: two species, two routes, and three dose levels. So if you think like suppose it is rats or mice, they have a short time period of reproduction. But when you talk of monkey or dog or other, then it will take years to reproduce and mature, again reproduce, like that. And carcinogenicity, the requirement is 2 years. So you have to monitor the drug, administer the drug for 2 years, and again same way, and observe for any carcinogenic effect. You have to monitor it for the carcinogenic effect. That is there any carcinogenic potential in the drug, any development of abnormal growth in the body or not? And coming to next is mutagenicity. Mutagenicity is the basically toxicity for genetics. That is there any toxicity or harmful effect of the drug on the genes? So what is the genetic mutating potential of a drug? That has to be examined. Again, in the same way, you administer to various animals, microbes, and study the effect of those on the genes. So you can think how many scientists are involved. As I told you earlier, hundreds of scientists are involved. You need genetic scientists, you need molecular biologists, you need chemistry scientists. So different types of scientists are required. Then, when we talk of dermatological toxicity, what is the toxicity in the skin? Ocular toxicity has to be studied, which is including that what is the effect on the eyes. So various types of toxicity study requirements are there, and all these requirements are laid down in the guidelines as per the guidelines of ICH and as well as CDS-CO in case of our country. So all regulatory bodies have laid down the toxicity guidelines and detailed procedures to be followed. So there's those detailed procedures need to be followed and adhere to for filing your IND. Basic purpose is that when you ensure and follow all these guidelines, you will be compiling the data and submitting it in the form of IND to the regulatory authorities. Now, some limitations of the preclinical studies include toxicity testing is a time-consuming and expensive process. As I discussed, that it takes many years. Then, large number of animals are needed or required to obtain valid preclinical data. The cell and tissue culture in vitro methods are increasingly being used, but their predictive value is still a very severely limited value. So extrapolation of toxicity data from animals to humans is not completely reliable. For statistical reasons, rare adverse effects are unlikely to be detected. So the applications filed with FDA or CDSCO after completion of preclinical studies and prior to human testing is called as IND. You have to file the application called as IND, investigational new drug application, as I told in the first slide. So which includes various components in detail, which includes chemistry and manufacturing information of the new drug. Now, as the name indicates, chemistry and manufacturing does not mean it's a small thing. It is in volumes. You have to devise a new information for a new molecule with respect to what are the compounds that are required to manufacture or synthesize that compound, what are the steps, scheme of manufacturing or synthesis, scheme of synthesis we call it as. So different types of chemistry parameters, development of assay methods, and moreover, very important is the metabolites. The metabolites means once the drug is ingested into the body, it will break down into various metabolic or metabolites. Now those metabolites will be very new, unknown to us. So you have to isolate or take samples of those metabolites, then identify those metabolites and develop the assay methods. Now this is a very tedious task. You have to develop the assay of the metabolites or new metabolites, I will call it as, which are not known till now. Those metabolites are extracted out, and the regulatory requirement says that you have to find out or develop method for all metabolite that is formed. Suppose a drug, new drug is X, and it is after entering the body, it breaks down into 4-5 metabolites. Suppose X1, X2, X3, or X4 like that. So you have to find out and identify all those metabolites. You have to find out chemical methods to identify, to analyze, and do the assay of all those metabolites that are formed, which are unknown to us till now. So that is a tedious part. So chemistry information has to be there. Then manufacturing information in large scale, how it is manufactured. Is there any environmental hazard or any chemical which is going to damage or harm the environment? Is that chemical being used or not? That will be examined by the regulatory authorities. Then, of course, preclinical studies information, as we discussed earlier, all that information which has been collected during the preclinical studies. Now, it appears like a very simple thing, but when we compile this information, how much data this becomes, all this IND application is the volume of application, if we call it as, it is more than one room of documents, one full room of documents, or one truckload we can call it as. So one that much is the volume of documents which are being generated, and all has to be submitted and they have to be examined by the regulatory authorities. You can think of it how much critical work they have to carry out. So after submitting all this, you are going to get a permission for conducting the clinical trial. If it's subject to their satisfaction, it is approved or not approved. That. So the clinical trial approval is given, and then you get a permission to conduct the clinical trial. So when you get the permission, then we have to perform the clinical trial in the drug development stage, which is now a very upcoming field nowadays, because a lot of clinical trials are being conducted and required to be conducted for any drug, new formulation, whatever it is. So clinical trial is a systematic study of new drugs in human subjects to generate data for discovering, verifying the clinical, pharmacological, which includes pharmacodynamic and pharmacokinetic and adverse effects, with the objective of determining safety and efficacy of a new drug. The research studies involving people or humans, and it tries to answer scientific questions and find better ways to prevent, diagnose, and treat disease. Why clinical trials are performed? The clinical trials are performed because no chemical can be certified as completely safe. Every chemical has some toxic effect at some dose level. So that has to be identified. What is the toxic dose level? To ensure or estimate the risk associated with exposure to the chemical under specified conditions by performing appropriate tests. The clinical trial may be required before the national regulatory authority approves the marketing of a new drug or device. Even devices are classified as drugs. Devices means medical devices we are talking about. Medical devices or new dose or drug that is used on patients. Then why clinical trial is necessary is to assess the safety and efficacy. You can see the term written as safety first and efficacy later on. Rather than writing efficacy and safety, you have to see that safety is first, to assess safety and efficacy of a new chemical entity, assess safety and effectiveness of a different dose. If there is some already existing drug with a in the market, but if you want to alter the dose of a drug for a new disease or to be tried, then again you have to carry out a clinical trial on the different dose and establish the safety and efficacy, and then you have to get the permission for marketing the drug in a low or high dose as per the requirements or as per the study expected. Or clinical trials are also conducted to assess safety and efficacy of an already marketed medication or device for a new indication. Now this is the interesting part. Suppose there are so many drugs which are already available in the market, and you come across a theory or hypothesis that this drug X or A drug X, Y, Z, whatever it is, may be beneficial in some disease, other disease, which is other than the indication that drug is marketed for. So it is approved for a particular disease, but if you propose that this drug may be beneficial in some other disease, again that has to be established by conducting a clinical trial. The efficacy and safety has to be established in the other disease. Then other aspect of clinical trials can be to compare the effectiveness of in patients with specific disease of two or more already approved or common interventions for that disease. So this is some history of clinical trials. I just want to skip this because it will be very lengthy then. So I'll just talk about four components of clinical trials. As we know that it involves human subjects, the clinical trials, they move forward in time. They must have a comparison control group. They must have a method to measure interventions. They must focus on the unknown aspects, like specifically the unknown effects of a medication or new medication. There should be a great focus on that. The clinical trials must be done before any medication is a part of standard of care, and they are conducted early in the development of therapies. When we are conducting a clinical trial, it is very must and important to review existing scientific data and build on that knowledge. Test a certain hypothesis. Study protocol must be built on sound and ethical science. Control for any potential bias. So there should be steps and measures taken to control any potential bias, like randomization of the groups, blinding of the groups of patients and investigator or physician. They should be blinded. They should not be knowing it, which drug is being given to which patient. Most study medications, procedures, and other interventions are required to be examined. So there are different designs of clinical trials that are used, which include randomized blinded trial, randomized double-blind trial, non-randomized controlled trial, placebo trial, crossover trial, withdrawal trial. So there are different types. If we discuss one by one, in briefly I will discuss rather than going into detail. Randomized trials, as the name indicates, These are the methods that are adopted to randomize or allocate the subjects or patients to different groups without any bias, to assign the participants to a particular group without the choice or bias of a investigator or the physician. Non-randomized means this non-randomized means all are not randomized, but they are given by the choice of the physician, which are patients to be given as treatment cases and which are given as control group. Then different types of blinding is done. In this, there is a single blind and double blind. In a single blind or normally called as blinded study, the participants do not know whether they are in the experimental or control group or placebo group. Whereas in double blind, the participants and the staff means the physician or the investigators, they do not know which group the patient is assigned to. So that becomes a double blind, and it is a very strong, robust design. Now, there is a term called as placebo. Placebo is a nothing but an inactive pill which resembles in size, shape, color, appearance with the therapeutic drug, but with no active constituent into it, so it does not have any therapeutic value, but only the excipients are there. It resembles with respect to color, shape, size, appearance. So some more components of the clinical trials include is that there should be at least two groups, minimum two groups has to be there in clinical trials. That is, one is the treatment group, second is the standard or placebo group, which we call it as control or placebo group. So it can be called as standard control or placebo control group. And there has to be a specific inclusion and exclusion criteria, very important aspect, specifically laid down inclusion and exclusion criteria should be there. The sample size of the clinical trials should be calculated, that how many number of patients the study has to be carried out by using various formulas are there, by power calculations. Then you should plan regarding potential bias, where the bias can occur with respect to screening of patients, with respect to inclusion of the patients, with respect to allocation of the patient to different groups, like that. And then also there should be a plan or a mechanism to handle attrition. Attrition means loss of follow-up. Suppose you want to carry out a clinical study on 200 patients or 300 patients, and 30 or 40 are lost at the follow-up. So it will lead to a loss of study, and the study cannot be completed. So there should be a mechanism to recruit some losses, or to cover the loss of follow-up, there should be some extra recruitment. So when we recruit the patients, what are the criteria or points to be kept in mind is, in clinical trials, identify eligible participants. Very important that once you lay down a inclusion and exclusion criteria, that criteria has to be followed. By that you can identify which are to be included and which participants are to be excluded. Like, for example, you are carrying out a study on new drug for hypertension or diabetes or whatever it is. So you will lay down a values that who all fall in that range will be included, and who all fall out of that particular range will be excluded. Or in addition, if somebody is having some other disorder like some hormonal disorder, some other concomitant disease, and taking some other concomitant medication, they should be excluded so that it does not interfere with your study. Likewise, identifying eligible patients is very important. Then you have to explain the study to the participants, which is called as informed consent. That means you have to explain and inform the participant about the study, and take a consent from the patient or the participant regarding participation in that study. It is very important and mandatory by regulatory authorities that you have to give a written informed consent. You have to give a written informed consent. So as the name indicates, it is a written document, and informed means you are informing the patient or the subject regarding the trial or research study and getting a consent for the same. So that consent constitutes written informed consent. So that information in that consent, all the information is there, like what this is a research study, this are the new drug, its background, what are the other options available, what is the duration of study, how long it will be carried out, and what is the time one has to devote. One has the freedom to participate and dissociate from the study without assigning any reason and without any compulsion. So, like that, rights and safety of the subject is maintained. Then again, care should be taken to reassess the eligibility and assigning the patients to one group, one or the groups. So once the experiments in the lab or preclinical established, then it's safe. Then you have to go through different phases of clinical trial. So different phases of clinical trials, as you are aware of it, it includes phase 1, phase 2, phase 3, and phase 4. That is phase 1, which includes healthy volunteers. Now, why healthy volunteers? Why do we need to study on healthy volunteers rather than we can study on patients? Why we have to go for phase 1 instead of this? We can start with the patients itself with small dose or small number. The basic purpose of carrying out phase 1 is to establish the dose range finding studies, especially the pharmacokinetics, that what is the pharmacokinetics in a healthy volunteer. Then only you can assess the dose, what is the suitable dose or appropriate dose which will be used in humans, and does it have any effect on the healthy persons or not, on various functions like liver function, renal functions, or on the blood parameters and all that. Various parameters are examined, and then if it is safe and efficacious, safe basically in healthy patients, then only it is given to the patient volunteers, which is small in number, as phase 2. And once it is successful in those small number of patients, phase 3, which includes large number of patients. Normally there are few hundreds of patients in phase 2, and whereas in phase 3 there is thousands of patients. So once the drug is established as safe and efficacious in phase 3, then you go for applying for a application called as NDA. If you remember the first slide, that is if you try to remember the first slide, the filing of NDA. So filing IND and filing NDA after phase 3. So once you are through with the phase 3, you go for applying for a NDA, and that is the new drug application, and you get a license to market the drug or approval of a drug, and you can market it. But again, still there are requirements that after getting approval, you have to monitor it for 2 years to 5 years, depending on the type of drug. The norms are varying. So post-marketing surveillance studies has to be carried out. When we talk of clinical trials, there are 3 basic ethical principles which has to be kept in mind and which has to be ensured that they are not compromised. That is respect for persons or subjects. That means treatment of any trial subject or person as autonomous. It's very important that you have to give full autonomy to the trial subject. That means the trial subject has full freedom and autonomy to take the decisions regarding with respect to participation or dissociation from the clinical trial without any compulsion and without assigning any reason. The second is beneficence. This includes the issue, that means you have to assess any potential conflict between good of society versus individual. That means you cannot compromise with the benefits of an individual for the sake of getting the scientific knowledge or talking of large benefit of the society, but if the individual is harmed, then it is not beneficence, so that should be taken care of. Then justice, that you treat all the subjects fairly and equally to share the benefits and risk. So there are some principles of good legal practices for conducting legal trials. So I don't want to discuss in much detail because it will be very lengthy, but briefly I will just go through that they have a background which originates from Declaration of Helsinki. You must have heard Declaration of Helsinki. Helsinki is a city in the country Finland in Europe. So in 1964, it was laid down the guidelines. When we talk of history of the legal trials, they laid down some guidelines termed as Declaration of Helsinki, in which the rights and safety of the well-being of the trial subjects were protected. That you should protect this safety of the trial subject. So in this, before any trial is initiated, foreseeable risk and inconvenience should be weighed against benefit. The rights, safety, and well-being of the subject should be important or most important. Then there should be scientific, there should be non-clinical and clinical information scientifically available for the new investigational drug, which should be adequate to support the proposed clinical trial. Very important. The clinical trial should be scientifically sound and described in a clear, detailed protocol. A trial should be conducted in a compliance with protocols that have received the Institutional Review Board or Institutional Ethics Committee approval. So whatever is there, in case of animals, you have a ethics committee called as the Institutional Animal Ethics Committee. In case of humans, it's called as Institutional Ethics Committee or Independent Ethics Committee. So there are two ethics committees in a society or the institutions. One is Animal Ethics Committee and Human Ethics Committee. The medical care given to the subjects should be equally of equal quality or high quality as given to the normal patients, and is the responsibility of the physician. Every individual conducting a trial should be qualified, then freely informed, freely given, informed consent should be obtained. Then all the data that is generated from the legal trial should be accurate. It should be accurately reported, interpreted, and verifiable. There should not be any ambiguity in the legal trial data. And very important aspect is the confidentiality of the trial subjects, especially the trial subjects and the records which can lead to identification of the trial subjects should be protected, respecting the privacy and confidentiality rules governing the different countries. And the investigational product, now very important. When we talk of this clinical trial, we say or we may think that it's only a small population. Well, let us insert a new drug and examine it, but it has to be synthesized or manufactured in accordance with the GMP norms, as per the standard GMP norms, and all the systems to assure the quality of a trial should be implemented. So this is summary of different three phases. Phase 1, 2, 3 includes healthy volunteers 20 to 100. Phase 2 as told few hundred, and phase 3 is few thousands. The purpose of phase 1 is safety and kinetics. Phase 2 is short term or small number of people for effectiveness, and phase 3 also. Now after phase 3, getting the approval, you have to perform post-marketing surveillance. After treatment has been approved and is being marketed, evaluate the side effects that are not apparent in the phase 3 trial. Thousands of people are involved. So what is an institutional ethics committee? Another small aspect. Briefly, I'll try to go through this. There is an IRB or IEC, which is constituted for clearance for any clinical studies. It conducts periodic review or gives a approval for conduct of the biomedical research, which involves human subjects. There are guidelines laid down for this by ICMR. Biomedical research involving human subjects. Guidelines for biomedical research. Responsibilities of the ethics committee is to review research to ensure that the potential benefits are outweighing the risk. Develop and issue written procedures. Review research for risk benefit analysis and proper protection of subjects. So the benefits must outweigh the risk. Always the written notice should be given for approval. Then the ethical committee should review and respond to proposed protocol changes submitted by the investigator if there are any changes, and review the reports of any adverse effects, serious adverse effects, death, mortality. If there is anything that should be recorded, and they should explore that. And continually, they should conduct periodic, continuing review of the study. Time to time, they should keep on conducting the review of study. Coming to informed consent, as I told you, that informed consent is voluntarily to be given. It should be comprehensive and it should be informative. The person is free to give consent to participate. They should be given by a competent person. It should not be given by a person who is not competent, by mentally ill children, like that. What is the components of an informed consent?
Hello, sir. We can't hear you.
I'm extremely sorry from the behalf of Dr. Pawan Kishan. Due to some network issue, he couldn't join right now, so he will be joining the platform shortly. So I request everyone to hold the line. I request everyone to hold the line for some few minutes. Thank you.
Sabadev, have you contacted with him on the phone line?
Okay, sir. Okay, okay.
Somebody.
I just share it. Am I audible now? Am I audible?
Yes, sir, you're audible and the screen is also showing.
Okay, so since when I was out of network or out of the meeting, I can re-continue again if you feel like. You want me to repeat some last slides? Do you want me to repeat the slides? Few slides if you have missed anything? Dr. Roy, am I audible?
Yes, sir, you're audible. Sir, you could continue in your pace.
Yeah, no, I'm winding up because I'm in the end. Because if you want, I if you missed something, then I will repeat. Otherwise, I am through. Thank you so much. Thank you so much. Dr. Roy and other members, am I audible?
I should really thank to sir for such an informative lecture, a very informative lecture. And I think that everyone, I'm sure that everyone in the meeting
Do you want to repeat?
their knowledge.
Do you want me to repeat some slides? Do
Sir,
you
I will ask to the audience. Do you want to have invitation? So request to any of the person that if you want, sir,
your voice is cracking?
slides.
There's some network issue. There's some network issue at my end. I think.
Yeah, yeah.
So
So is it fine now?
anyhow, apologies for the network issue.
Sir, is it fine now? Can you hear me?
Yeah, I can hear you, but it's cracking. The voice is cracking, breaking in between. Anyhow,
I would request to
am I audible clearly?
request to have queries. If you want the survey slide, then you please raise your hand or you please be on the microphone. To all the
Thank you.
participants, if you want to repeat any slides,
Yeah, you are welcome to ask any queries or questions. All are welcome. The
and
participants
I
are
request you that you please ask to any query
yeah, well, participants are
relevant to this
welcome.
lecture.
Participants are welcome to ask any queries or questions or clarification they need to ask. They are welcome. Yes, if there's anyone who
I should ask any questions. Somebody, if you have any questions, you can ask. Saurav, Shivaji, any questions or participant from other colleges?
Sir, can you please explain the lead optimization process once more?
okay, basically in lead optimization,
Yes, sir.
it is it is a refining of the properties of basically it is with basically it is with refining of this disturbance.
Okay, sir.
Hello,
Hello.
kindly mute it. Means basically the lead optimization means basically it is with respect to the refining of the properties. Am I audible? Am I audible?
Yes, sir, you are audible.
Okay. So basically, lead optimization means it is with respect to refining of the chemical or physiochemical properties of a drug. That means how we can improve the binding or how can we improve its absorption or various types of its binding with the target or a target molecule. So various properties to enhance the properties, various structural changes are carried out so that their uh, this thing physicochemical properties are improved with respect to solubility, binding, so that there's improvement in the efficacy of a drug or to decrease the toxicity of a drug. Various types of studies are carried out or experiments are carried out.
If any of the faculties are having questions, ask.
The voice is not clear, pardon.
If any of the faculties from the group want to ask any question, they can ask.
Yeah, sure. You're welcome. You're welcome to ask.
We have got any from anyone, and sir, lecture so informative. So informative. I don't believe. So anyone should be having this, because I should say, I shall repeat the same thing, that sharing the lecture with sir, just listening the lecture with sir, is like to pick up the particles from the seashore. Sir is an eminent biologist. To listen to the lecture is like to pick the drop from the sea. So whatever the knowledge he's having. So with that, I shall really thank you, sir, for having his valuable time and to spend a long time and make our activity very, very successful.
Thank you.
So thank you so much for your timing.
Thank you so much. The voice is
Thank you so much, sir, for your participation, for your joining, and for to do the activity.
Thank you, sir. It was a very insightful FTP.
Thank you all of you. Thank you all of you and best wishes. Dr. Roy, is there any other queries?
Thank you, sir.
Dr. Roy, is there any other queries are there or? Yes, any other queries are welcome, please.
Yes, sir.
What can somebody ask clearly? I'm unable to hear the voice.
Someone have any? We are going to close the ceremony. Do somebody have any other question?
Yes, you are
Do
welcome.
somebody have any other question?
Yes, you are welcome. Any question? That will be a simple question, any whatever it is, any question. I hope you have got some insight into the drug development process.
Sir, I believe it.
I hope it
Sir,
was
I believe it
I hope it was beneficial.
from your descriptive lectures and from your explanatory lecture, everybody is very fruitful.
Okay, fine.
We are getting a lot of positive responses from the participants, and it is really an honor for.
Actually, we can have everything, every other topic in this have much more details, but this was an overview of regarding the process. Every step involves a lot of detailed steps are there. So thank you, Dr. Roy. If there is any query, if there is any question or query, you are most welcome. If there is any or if there is any question or query, you are most welcome. Otherwise, we can wind up.
Yes, sir. I think so that nobody is having any other questions.
Okay.
So with that, I'd like to close the ceremony, and I'd like to thank Sir a lot for his valuable timing.
Thank you so much.
It's a really an honor to hear from you.
Okay, thank you, Dr.
Thank
Roy.
you, sir.
Thank you, all of you.
Thank you, sir. Thank you very much.
Okay, thank you.
So wishing you
Thank you, sir.
future.
Okay, all the best wishes to all of you.
Thank you, sir.
Dr. Roy, thank you so much.
Thank you so much, sir, for your valuable timing. And it's a great honor to have you over here.
Right. All the best wishes to all of you.
And in the near future, also, we are planning to bring you over here.
Take care. Be happy. All the best.
Thank you, sir. Thank you.
Okay, dear.
So all can leave the meeting?
Shall I leave?
Yes, sir. Yes, sir. Please.
Thank you. Thank you, all of you.